My name is Barry Williams. I'm a faculty in zoology and microbiology here at Michigan State, and I'm going to be presenting a very general overview of some work that I've been doing in collaboration with Heather Easton and Toria Davis, who's employed by Beacon and works in both of our labs, as well as Louise B, who's been working on salamanders and Sodefrin long before she came here. So I'm trying to keep this as general as possible because I know the audience is quite general here. And what I'm going to be talking about today is sort of a little bit about what we've learned about molecular evolution of a gene. And so I'm telling this story exactly in reverse from how I learned it. And when we started out, Heather came to me with this great idea. She had a behavior and a gene responsible for that behavior. Both were very interesting. And as I tried to figure out the origins of this gene, I found out that I was inept and I needed help. Toria, Heather, and Louise have been a great deal of help, Toria in particular, in getting a lot of this work done. I'll be presenting some of the generals of it today. So, the gene that I'm interested in, that we're interested in, has a domain that's called a three-finger domain. Well, what's a domain? Domain is part of a protein. If you'll remember central dogma, you have DNA with, on that DNA, if there's a gene, a simple protein coding gene, there will be exons and introns, there will be untranslated regions. RNA polymerase will come and make eventually a pre-mRNA. The introns will get spliced out. You'll end up with a mature RNA with a polyadenylation signal and a 5' cap. That RNA then gets sent out. It goes to the ribosomes where it gets translated into protein, which I've shown here with a very simple blue rectangle. Within a protein, proteins can be, but are not always, composed of domains. Proteins can be quite modular, in fact. Different genes can be composed of just shuffled domains, exchanged among various genes. So when we look at genome evolution, one of the things we're interested in is what gene families have expanded and contracted, because maybe whatever that gene family does is a particularly important thing. Similarly, as we get better and better at understanding protein domains, we've started to examine what sorts of domains have expanded and contracted over evolutionary time across taxa. And the reason we're better at finding domains is because there are much better algorithms for searching for evolutionarily conserved protein sequence, protein structure, and we're better and better at predicting protein function. So these domains can often be isolated, separate from the rest of the protein. They will stably fold on their own, and they still exhibit the same biochemical activity that they would in vivo, even when they're expressed in vitro as a domain on their own. So I started by telling you we were interested in a gene, but okay, go ahead. What's the question?
Someone needs to mute their microphone.
Thank you.
Me? Should I mute mine first?
I'm enjoying this.
We're getting a lot of noise from one of the other institutions. Could everybody please make sure your microphones are turned off? Thank you.
Okay. All right. So domains, little parts of proteins can act on their own, can be shuffled around. All right. This is called a three-finger domain because it makes, not surprisingly, what are considered three fingers. Really, there are three loops. So whenever we find a three-finger domain, the only thing we know about it, it has a series of cysteines. That's the amino acid. Two cysteines come together, will form a disulfide bridge. Those are very stable. They tend to be internal parts of proteins. I hope you're not falling asleep yet. But it's important because that's what's holding this domain together. That's the glue. These cysteines are spaced across the domain so that when they come together, they fold and create these three loop structures. And those have been termed fingers other structures in structural biology are called fingers, and they're not simple loops like this, but in this case, it's actually pretty simple to understand. This domain is only about 70 to 100 amino acids in length, sometimes as long as 115 amino acids. But it's basically composed of a series of cysteines, and whenever we find it, this is the structure. Compare the structure among different three-finger domains from very different organisms with very different jobs to do, and they look like this. And it's a pretty simple structure, so it's kind of hard to get it wrong. Now, what does the sequence itself look like? What I'm showing here in the alignment you see at the top are 5 closely related snakes that I'll come back to. Those snakes contain a gene that has this 3-finger protein domain in it. These are closely related snakes, but what I hope you can see is that other than the highly conserved cysteines, which I've highlighted for you, everything else has already been scrambled. Any other gene would have hardly any variation whatsoever. This thing's going pretty fast. And probably the most interesting thing about 3-finger domains is when we find them, no matter what organism we find them in, they're evolving incredibly rapidly. And so far, every test that anyone else has done, along with the tests I've been doing in my lab with Toria, comes up showing that these things are evolving so fast they can't be explained by neutral evolution. It's not that they do nothing. The amino acids are changing so fast that it would appear amino acid substitutions are preferentially fixed in an adaptive fashion by natural selection. We don't know why, but every time we find 3-finger domains, no matter their job, they're evolving rapidly, with the exception of these cysteines. So the next thing I'm showing is one of the big advancements that's happened in domain searching algorithms. It's called HAMMER. It's a hidden Markov model way of looking for sequence patterns. It's particularly good at finding domains. And what I've shown here is for 3-finger domains. This represents the hidden Markov model that describes sequence variation across all 3-finger domains that have ever been found. There's a lot of them. I'll come to that in just a minute. And what I hope you can see is the height of, okay, so each column here represents a position within the domain. So we're just reading protein sequence, amino acid by amino acid. The height of each amino acid within a column indicates how important that particular amino acid is in this search. So this cysteine is pretty important, and no other amino acid seems to be important. Over here next to this cysteine, we see a Y and an F towards the middle. So that says that the amino acids, the symbols here don't matter. We'll just stick with Y and F. Those 2 amino acids are both fairly important, but they're not that important. Added together, they're not that high. Over here on the right-hand side, the C-terminal portion of this domain, you can see that there's a double cysteine and then another C followed by an asparagine and N. This is what a 3-finger domain protein looks like. A bunch of Cs, a bunch of cysteines. 9 to 13 cysteines within 100 amino acids. The nice thing is that doesn't happen by chance. That's actually pretty hard to find. Because cysteines, when they come together, tend to form disulfide bonds, evolutionarily, they don't happen in pairs very often. Proteins tend not to be cysteine-rich, and when they are, they certainly don't have this kind of spacing pattern. So we can find them, but this algorithm often fails. So some of the very first 3-finger domain proteins discovered by biochemists, their structures were described, are not found by this algorithm. The algorithm is designed to find the thing that it can't find, but it finds other ones. So a lot of our ability, yeah, isn't that good news? A lot of our ability to find 3-finger domains these days is obviously through sequence searching, not through biochemistry and function. So here's another way to look at 3-finger proteins. Along the top, this is another alignment of genes within a species. At the bottom, by the way, there's what look like 3 taxa. That's actually one gene for humans that's composed of 3 3-finger domains, one after the other. Even those different domains, the cysteines align, nothing else does. Some people have decided that we shouldn't call them three-finger domains. Every time they get discovered, and they've been discovered many, many times, they're given a different name. Whoever finds them in a particular taxa gives them whatever name they think describes what it does. So some groups, I found this paper in Fed's Letters from the mid-90s to be particularly helpful. They broke up different three-finger proteins based only on the cysteines and the spacing pattern, but you can see if you look at these numbers, it says, well, between these two cysteines here on the top one in the middle, there's somewhere between 18 and 24 amino acids in there. We don't know what they are, but it'll be about 18 to 24 amino acids. And so based on these spacing patterns of these cysteines, we can start to maybe categorize these things a little further. This falls apart. Okay, there's lots of three-finger proteins that don't look like this. Again, some of the first ones discovered don't always look like this. It's a general rule of thumb, but when we're looking for three-finger proteins, this is what I spend my days doing. I'm looking at a screen, Toria's looking at a screen, she's rolling her eyes at me often. She's trying to find cysteines.
She's upstairs in my lab complaining about spending all day looking at a screen.
That's right. Looking at letters all day, trying to find the letter C in a particular pattern. And I'll tell you what, if you show her a bunch of letters on a page, and the C's match this pattern, she'll instantly know if it's a three-finger. So she's quite good at it. I'm sure she'll get a job because of that. Where do we find three-finger domains? What species have them? Animals have them. There's only one case of ever finding them outside of animals, and that was a herpes virus that infects animals. So it may be a case of horizontal transfer. But despite the many genome sequences that exist outside of animals, obviously, it's never been found before. And again, it's pretty hard to miss. So if we, and I mean all animals appear to have this thing. It originated with animals. It's in all the metazoa. But when we find it in metazoa, this sort of table is taken right from one of the websites that searches for these things. And at the top, you'll notice, ooh, that's the wrong button. Okay, at the top, you'll notice metazoans. There are 265 species. So the number of species that are known to have this domain are listed. The number of genes within those 265 species that have at least one is about 2,500. So roughly 10 genes per animal contain a three-finger domain. And as you can see, this 2,900, that's the actual number of domains. It's bigger than the number of genes. So genes often have more than one three-finger domain within them. Here's what's interesting. If we look at annelids, mollusks, nematodes, even arthropods look to have somewhat of an expansion. If we look at 32 species of arthropods with full genomes, there are 167 genes that contain three-finger domain proteins. So if we do a little bit of division there, roughly 50, right? So there's going to be roughly, sorry, not 50, that's 5, roughly 5. And there's 168 domains, so each gene contains one domain. So given all animals have them, and most animals don't have many, there's got to be one group that has a lot. That's what's at the bottom. Chordates. Out of 206 species, they have 2,284 genes that contain these domains, and there's more than 2,600 domains. So in fact, even though all animals have them, almost all of the three-finger domains are found in chordates. Let's go a little further. Within chordates, by and large, most of the copies are found within tetrapods. Now, we don't know why, but it's pretty unusual to have a gene or a domain that explodes in copy numbers and is associated with anything in animals, because by and large, animals have very similar numbers of genes. There are some expansions, but to this extent, it's pretty interesting, actually. So for a domain that you may never have heard of before, this thing is doing something special. Within the teleosts, okay, so here we're talking about the fish, there are 16 genomes, there are 232 genes that contain a three-finger domain protein. So this is where we're getting up more than 10. But when we get to humans, okay, there are 93 genes that contain three-finger domain proteins. And 93 genes, but 107 domains. As I said, gene copy number tends to evolve slowly in animals. So if we look at mouse, human-mouse comparison, same number of genes, 93. Within the plethodons, salamanders, there are 28 genes, roughly, per species. And within Xenopus, that we've now found, the scientific community has found 42 genes that contain this. So there's a pretty biased distribution of these. They're only found in animals, largely found in chordates. And within the chordates, they tend to be much higher copy within the tetrapods. There aren't many things within genomes that have this kind of explosion associated with them, only found in tetrapods. Okay, so I'm talking about the molecular structure, and I promise to stop doing that. So now I'm going to ask, what do they do? Why are we interested in them? First of all, I want to tell you they come in two flavors. Either, once they're made, they're attached to some kind of gene that gets sent outside of the cell and gets stuck on the cell surface and does something there. And I'll show you that something in just a minute. Or they just get sent out of the cell and then they float away on their own. Not going to get stuck to the cell wall. So they're just simply secreted. What kinds of genes are they? Well, they're genes I'm sure you're familiar with. You've heard of complement regulatory proteins and lymphocyte antigens and plasminogen activators and urokinase receptors and phospholipase inhibitors. I'm going to tell you that you're going to be interested in them in just a minute. Okay? It's a long list of genes. And even though all of these genes is largely composed of a three-finger domain, they come up with different names for this domain every time. So some people would call this domain a Li6 domain, or part of the Li6 superfamily, because it's one of the lymphocyte antigens. Lymphocyte is the Li, it's the sixth antigen ever found. Some people call it PLI, that's for the phospholipase inhibitor. Some people just call it a toxin. So what in the world's going on? How can this one thing be in so many different types of genes? I think that there is a common link among these genes, and this is where we get to my cute little title about potions, poisons, and perfumes. And I'm going to actually try and go through this one by one and demonstrate that I think that these three are actually good descriptions of what's going on. The first is the way that three-finger domains were first discovered. And that's because snake venom, one of the neurotoxins that they contain, is a three-finger domain protein and nothing else. It is simply a secreted three-finger domain. So they're tiny little proteins. And they specifically attack acetylcholine receptors. And this three-finger domain is what was one of the first things that gave us the molecular tools necessary to study how receptors bind ligands. Here's one of the first ligands with a known receptor. All you need to know is that if it does bind your acetylcholine receptors, you will die a painful death very quickly. So I think that's a pretty good qualification that this domain is a poison. Snakes make it, they produce it in their liver, it gets sent out as a toxin, and then, well, it's just purely a poison. This one's a little weaker case. I'm definitely stretching here a little bit, but there's one called xenoxin. Now, I was just out there looking for a video. There are videos of frogs. It was too long and it was a bad video, but there are videos of frogs escaping the jaws of a snake because their skin secretes bad things. Their skin can also secrete antimicrobial peptides as well. And so some people, in studying what is secreted by the skin of frogs, like Xenopus, the main lab animal in the frog world, found a three-finger domain. And their logic was pretty simple. Frog skin can contain some nasty things. This is a three-finger domain. That's what snake venom is made of. This is probably something related to a venom or a toxin. It's a poison. It's a poison from Xenopus. It's a toxin from Xenopus. So Xenopus toxin is shortened to xenoxin. They then tested it for toxic effects, and it had none. That didn't cause them to change the name. However, They worked a lot longer because they had kind of been scooped, and it would appear that they found that this thing may actually activate specific receptors in neurons. And that is interesting because it gives a novel case of a receptor-ligand pair that wasn't previously known. By the way, this is very much Heather's domain, and I'm saying very little because if I say anything, I know it's going to be wrong. And I don't, I'm sure I've already made a lot of mistakes, and I'm just trying to reduce the amount of trouble I'm going to get into after the talk. Okay, here's another reason where I'm stretching even further that they're a poison, but they're bad news within your body. Your genes are doing things with three-finger domains that you don't want them to do. So those urokinase receptors I was talking about, urokinase receptors bind plasminogen. So when they do, plasminogen, you can think of it kind of as a molecular drill. What it can do is it can chew through the extracellular matrix, but it is not activated until it binds the receptor. The reason that you might be concerned about this is what I'm showing here in this image is this is cancer cells basically becoming malignant and migrating through an open pore. Cancer tumors can't move often within your body because they are, except for things like lymphoma, they are often embedded within tissue. And so that tissue holds them in. So for cancer to become malignant, for metastases to form, the first step that has to take place is you have to activate your urokinase receptors and activate plasminogen. And basically they've now got a drill. And these cancer cells can drill their way out of the tissue and become metastatic. So if you have cancer and for whatever reason your tumors start expressing this particular gene at high levels, it is, well, it's not poison, but it's really bad news. It's the strongest signal there is today that you're probably going to have a metastatic tumor. Now, Ly6, I said lymphocyte antigen number 6. It's really interesting. This is part of our MHC. These three-finger domains are one of the reasons that our MHC is so variable. It's because there's lots of them found within there, and there's lots of copies of this thing. And we don't really know what it does. And so this is like an antigen. We found that our T cells are producing this thing, and we can get antibodies that detect this thing, but we don't know exactly what they do. If we take them away, it appears that that influences cell differentiation. But the reason I have it, so that's part of our immune system development. The reason I have it here as a poison is a lot of viruses, including HIV, and more specifically, it appears adenovirus, use the Ly6 receptor area as a point of attack. That's how they get into your cells. So, when they do, by the way, HIV, the virion often picks up pieces of Ly6, and that's what it presents to your cell to escape immune function. So they use it against us. So three-finger domains are doing a lot of things in a lot of different organisms already, but I'm going to transition out from poisons to potions. And the first potion I'm going to talk about is we're going to go back to snakes. So even though they're making three-finger domains as a poison, snakes are actually immune to their own venom. And one of the reasons they are is there's a couple of main toxins they make. The other is this enzyme called PLA2. All right. It's an enzyme, and it attacks cell membranes. It lyses cells. Well, in their blood, they're synthesizing PLA2 inhibitor. PLA2 inhibitor is just a three-finger domain protein that's floating around in the cell. So they're making two kinds of three-finger domain proteins. One will kill people, and the other will save them. One is a venom. The other is an anti-venom to another form of the venom. So here's something that's going to save lives. It does often. Are these PLA2 inhibitors. Again, all of these three-finger domain proteins, when we compare them among species, these loops are evolving incredibly rapidly, so it would appear they're under natural selection. Another form, another name that people give three-finger domain proteins is CB59, or UPAR, and that goes back to the urokinase receptor. do you see how many silly names I've given these things so far, CD59, Y6? I think one of the reasons that putting these things together is that they're not often called three-finger domain proteins. Everybody refers to them within their little sub-discipline. So people who study CD59 don't always know that they're studying Y6, don't always know that they're studying UPAR, don't always know that they're studying toxins. But CD59, that CD stands for complement deficient, but this inhibits, normally within your body when it's working properly, it inhibits complement-mediated lysis. So it stops cells from dying. So here again, you make things, one of them is called Pinlet, but you have your own version of three-finger domain proteins that are used to lyse your own cells. And CD59 can stop that. It's a receptor, again, it's part of a regulatory cascade. And it's a therapeutic target because if tumors start expressing this at high level, then treatment to destroy that cancer fails, and you become resistant to chemotherapy. And so usually when there's chemotherapy resistance, it's because this gene is being highly expressed and including its ligand, which is also being highly expressed, which makes this things that will act as ligands for CD59 then are therapeutic targets. And there are many drugs being synthesized that are trying to do just that, okay, to dock at CD59. And as I said earlier, UPAR, this is the receptor that's going to hold on to what's basically a molecular drill. Again, for the same reason, this is the target of many cancer drugs. If we can find drugs that will bind to that receptor as well, then that can slow the probability or reduce the probability of metastatic cancer. I see I'm already going over. So I'm going to end with the cute, lovely organism. Think of all the nasty things this three-finger domain protein does. And I really don't know anything about salamanders, but I did learn initially from Heather, but it turns out these things are actually kind of cute. I didn't know. They look like they're smiling all the time. And Louise has shown me some of these too. And, because they're in their lab or they're in their house and they'll smile and say, "Oh, it looks great today. It looks really happy." And I just think it's just laying there. And they really think of these as cute. But if you look at them, I think they are cute. I'll give them that. And one of the neat things about them is they've taken this molecule and they're using it for love. It really is a perfume. So the males are excreting this, and I think I have, the males are excreting this from various portions of their body. It's a peptide, but it acts as a pheromone. Females detect it. They literally put their nose up to the male where the pheromone is being excreted, and then that's part of either the mating ritual or it can be used to guide the female, so that the females are more efficient at picking up the spermatophore that the male leaves behind. There are two forms of three-finger domain proteins. One is called plethodon mating factor, because it's only been found in the plethodons, and the other is called Sodefrin. This was one of the first protein pheromones. It was the first protein pheromone found. So they started by sort of doing forward brute force approaches, where they took stuff secreted from the cloacal gland of a red fire-bellied moon. Is that right? Red fire-bellied moon? And they figured out what was there. They took fractions of it, presented it to females. One fraction was particularly effective. Then they did biochemistry on that, figured out what it was, found it in the genome. And it's a gene called Sodefrin. Now, here's where it gets interesting. What I say is they're really cute, and look what they're doing with this gene. When we look at the evolutionary homology, and this is where it gets a little tricky figuring out where these things copies come from, but I think Tori and I have a pretty good handle on it. It turns out that these potential poisons, these xenoxins, that exist in frogs and other things that are excreted by frog skin that potentially act as antimicrobial agents are the homologs of Sodefrin and PMF, respectively. So it would appear that they took what is being used potentially as a toxin on frog skin, and they're now using it as a pheromone. And it's all salamanders appear to be using Sodefrin at least. So we really don't have extremely strong evidence that that's the case. I do want to show one thing before I'm done. And so this is just a video that comes from, this is from Steve Arnold's lab. So the picture I showed a second ago shows the red fire-belly newt, and this is the cloacal gland of the male. It's fairly large, and this is where the three-finger domain is being excreted from. But in plethodons, they have what's called a mental gland, and this is beautifully named because the mental gland occurs on the chin. So I don't know who came up with that, but instead of females going up to the male's cloacal gland, females actually come up closer to the males. And the males appear to distribute their pheromone in this case by sometimes slapping. Is that right, Louise? That these would be the ones that would be slapping against the... Yeah, presumably to more effectively present this pheromone. And then this is just showing, eventually once in the red fire-belly newts, the females sort of follow the males. I had a video of that, I don't know where it is now. But here in this species, once this process is taking place and the pheromone has been transferred, then the males will transfer spermatophore to the female. And this is part of this whole tail wagging and the female rocking back and forth behavior that sort of finishes that process. And these, in plethodons, these ratios of PMF to Sodefrin to some other chemicals, there's the spermatophore. Now the female is going to move over that and pick it up. So the rate at which she's receptive, the rate at which this transfer takes place is all influenced by these pheromones. But in Cynops, it's incredibly strong, the effect. Okay, so the last thing I'm going to leave you with today is I want to draw, this is actually, I know what this might look like, but it's actually sort of a protein representation of what this thing is made up of. So Sodefrin from Cynops is quite simply, the first 20 amino acids are just nonpolar amino acids, and that's a secretion signal to get it out of the cell. Then this blue area represents the three-finger domain protein, which is about 80 amino acids. And then unique to Sodefrin, there's another about 80 amino acids that are actually unstructured. No matter how you model it, you can't get it to fold stably. So it just sticks out there. And it turns out that what Cynops does is it cuts off basically the last 10 amino acids of this protein, and that is the peptide pheromone. So this three-finger domain, that's not even used as part of the pheromone. That's not what's being transferred. However, in other salamanders, it would appear that they don't cut off those last 10 amino acids. They use the whole thing. And so we're trying to figure out now which species would be most easy to work with, so that Heather can do some real functional biology and understand how things work. And the thing we're struggling with at this moment is when you sequence the genome of any one salamander, as you probably figured out, there's more than one of these things in there. But even if you're looking specifically for Sodefrin, not just any three-finger domain protein, specifically for Sodefrin, within one species here, the first 5 sequences are all from the same species. You can see that there's at least 5 copies of this. So now within some other species, people have estimated that there's up to 12, maybe more copies of this single gene. And so which of those copies is the one that's most effectively expressed in the tissue of interest? Which of those, or do all of those potentially act as pheromones? And are they clipping off these last 10 amino acids? There's pretty good evidence from the plethodons of distantly related group of salamanders that they don't cleave off any of those amino acids. So our goal now is we think we have a protein that's particularly interesting because it's a gene that creates a molecule that alters behavior that can be studied. That's hard to find. This particular molecule appears to be interesting in that it was co-opted, as it has been co-opted again and again, but in this case to do something particularly interesting. And because receptor targets are thought to be known in frogs, Heather may be able to directly go and find potential targets in salamanders, and having both the ligand and the receptor for behavior would be quite a breakthrough if we can get that. So I think that was my last slide, and again, I want to thank Heather, Tori, and Louise, and I'm happy to take any questions. Yes?
In surveying all these species where they've been using this, have you ever found it where they co-opted it for another function? So when you're talking, it made me think of sticklebacks that have this odorous nests that the young imprint on. It's clear when they're mating that the females are smelling something. And it's also secreted from the liver, like many of the things you looked at. Did you ever see that there was one where, a species case where that odorant is also used then for another function as well? Because fish are, I mean, they're much older, so it's possible that in that time they could have adopted another function. So perfume plus building material. Did you ever see anything like that potentially? Just out of curiosity.
So within a species, so that's more of a pleiotropic use of the gene, I think. Two jobs for the same thing.
Yeah.
And, I'm not thinking of a case within one species, but the potential here is that maybe in frogs or salamanders it is acting as both poison and perfume, because within frogs it certainly hasn't been tested as a pheromone, and within salamanders it hasn't been tested for antimicrobial activity or toxicity to the vertebrates.
It sounds super cool. Thank